So I wanted to make what I'm hoping will be a short video here to kind of run through the slides that we've had from our second and third learning unit. So our learning unit again on nucleic acids, and then our learning unit on protein structure, to kind of help you understand the important things that are essential to study for the exam. So one of the things I'm going to highlight is, and I encourage you to go back to essentially what equates to the first half of this sort of chart that we made in the last part of our introductory learning unit on sort of fundamentals of biochemistry. And even though this wasn't part of what you needed to know for the first exam, I kind of highlighted that this is sort of the high level stuff that I'd expect you to know kind of come the end of the course and so forth. So again, we've already touched on all of this with nucleic acids, and then again thinking about a good portion of this with protein structure. But go back and take a look at these, and all of this should sort of make sense as sort of the highlights of what we're going to talk about with nucleic acid chemistry and then protein chemistry. So I'm going to fast forward here to kind of our third chapter here on nucleotides, nucleic acids, and genetic information. What I want to just do is kind of go through each slide and sort of highlight the things that are important for you to think about. So one of the things I told you that's important to be able to know how to do is draw a little dinucleotide. I'm going to tell you to draw a dinucleotide. You're going to have to know, again, I'll tell you whether it's RNA or DNA, but that's going to dictate what goes on this 2' position, whether it's an OH group or a hydrogen. Again, you're not going to need to know the structures of the bases, but you might have to write that this is a C, for example, here, and this is a G, depending on the sequence given. I want you to be able to identify what the linkage is in a DNA or RNA little polymer here. So again, here's the phosphodiester bond. I want you to be able to identify the direction that we have here for this little piece, this little dinucleotide here. The last piece is understanding that I'm going to tell you whether it's 5' or 3' phosphorylated. And if it is phosphorylated, you have to put a phosphate group on one of these positions here. And again, this is what the phosphate group looks like. It's going to be essentially this oxygen, and then P double bond O, and then single bond O is there. So again, be able to draw something that looks like this depending on the information that I give you. Other pieces that I need you to know is I want you to know who the purine nucleotides are, A's and G's, who the pyrimidine nucleotides are, C's, T's, and then U's are for RNA. So I also want you to know who pairs with who. So A pairs with T, G pairs with C, and then uracil, U, represents essentially the T analog that we see in RNA. Again, other differences between DNA and RNA have to do with ribose versus deoxyribose sugars. And again, we already sort of highlighted that looking at the structure over here. So that's pretty much the basics from this slide. You do need to know how to draw the structure of ATP. That is going to be a question there, so make sure that you're able to draw this guy, be able to identify which bonds are the high energy phosphate ester bonds. So again, phosphate ester here, phosphate ester here. That is not a high energy phosphate bond there. Some numbers to know about that: the physiological concentration of ATP is 5 millimolar. That's coincidentally the same number that I need you to know for glucose. We haven't talked about carbohydrates yet, but that's an important number. This is again a number that you needed to know from our previous learning unit, value for ATP, and that value actually comes from looking at the hydrolysis reaction of ATP. And know too that the reason that ATP is worth this much is because when we cleave either of these phosphate ester bonds, what we're doing is separating charges. Remember, there's a -4 charge on ATP, and so when we cleave either of these bonds, we're actually separating the charges there. We talked about pyrophosphate here and the fact that you've got lots of pyrophosphatase floating around in your bloodstream, and that helps to impart irreversibility to reactions, because any reaction that generates pyrophosphatase will convert that pyrophosphate into 2 inorganic phosphates. The Chatelier's principle will allow the pulling of whatever reaction was happening forward, because we're going to be removing that product. Last little thing to highlight here that I do want you to know, this is an important piece, is understand that phosphatases are enzymes that are going to remove phosphates from proteins. Kinases are going to be enzymes that add phosphates. So again, know the names of these two kinds and classes of enzymes, as well as understand the differences between when we would be either removing phosphates from enzymes or adding phosphates to enzymes. So we call these phosphosystems or dephosphosystem. And I do want you to be able to connect that a dephosphosystem is what we refer to as a fed state. We're going to keep building on this as we learn more things metabolically that happen in a fed state. So when we're in a fed state, we're going to have lots of phosphatase activity. We're going to be removing or dephosphorylating proteins. When we say need state here, I just want to clarify, this is actually glucose or energy need state. We can generally, just refer to it as need state, but specifically it's going to be for glucose or for energy. So when we're in that need state, we're going to be having kinase enzymes decorating a lot of other metabolic enzymes with phosphates. So we'd refer to that as a phosphosystem. So again, this is an important piece. Make sure that you understand this terminology, these classes of enzymes, and what falls into what category. So B vitamins, I want you to know there's 4 B vitamins that we've learned thus far. So again, to highlight what I need you to know for them, you're going to see something like, all right, I'm talking about the B vitamin that's responsible for doing electron sort of harvesting via hydride radicals. Okay, so what do you need to be able to know for that? You need to be able to know that that's FAD and or FADH2. Remember, FAD is the oxidized form, FADH2 is the reduced form, so you need to know the metabolite that is. I also want you to know the vitamin name. So the vitamin name is going to be riboflavin. I want you to know specifically the number of the B vitamin. So this is going to be vitamin B2. So that's the details that I need you to know for these B vitamins. Just to kind of highlight here, we've talked about glutathione a lot. I do want you to know glutathione is a natural antioxidant in your body. It operates, and we'll see this in our protein slides here in just a minute. This operates by utilizing disulfide bond formation chemistry. So again, this is what we see for FAD or FADH2. It's riboflavin and then vitamin B2. To kind of talk about the same thing for NADH, the metabolite name is NADH. The vitamin we can either call niacin, or if you wanted to use nicotinic acid or nicotinamide, but niacin is kind of what you'll see on your vitamin bottle, also called vitamin B3. So again, this would be, a B vitamin that's involved in 2 electron transfers via hydride. So be able to kind of describe the kind of chemistry going on, which is kind of what's listed up here, the metabolite name, the vitamin name, and then the vitamin number. Okay, and again, this is just sort of a big picture thing that we've already talked about. So fuels, again, are worth value. They have value or worth energy because they're held together with electrons. And so electrons being energy, if we can harvest them by doing some redox chemistry, which is what these B vitamins help us do, that's how we get energy from our fuels. So 3 B vitamins that we talked about in this nucleic acid chapter. The third one is vitamin B5. So again, the metabolite that we're going to talk about is acetyl-CoA. The vitamin name is pantothenic acid, and the vitamin number is vitamin B5. So what do we need to know about this? Again, acetyl-CoA is responsible for transferring 2 carbon groups. So again, 2 carbon groups through metabolism, and that's what we call this acetyl or this acyl group. So don't need to know really the structure of the of acetyl-CoA, but acetyl-CoA has this important thiol group at the end, which is why we'll often write it as CoA if it's sort of in its reduced form, or if it's acetyl-CoA, it'll have this thioester. And again, highlighting that thioester group is why it's a money molecule. So we are saving the energy of these 2 carbon pieces that we've obtained through metabolism by creating this high energy thioester bond. So again, highlighting that when we undergo fuel metabolism and we harvest energy, we are basically going to be taking the electrons and they're going to go to these 2 B vitamins, and we're going to be taking the atoms and they go to this other B vitamin, vitamin B5. Those 2 carbon atoms eventually become carbon dioxide as we go through processes like the citric acid cycle. So this is going to be the main focus of what we're going to talk about. We'll mention briefly the fact that acetyl-CoA can be used to add acetyl groups, and that can be used again for signal transduction or for regulating the activity of enzymes. But this is going to be a minor contribution. This is the main point that I want you to remember. So again, we talked a little bit about Human Genome Project, epigenetics. I do want you to remember that epigenetics is the ability of the environment to control your genes. And thinking about this 80-20 rule, basically your genes control about 20% of what goes on. Even though your genes really control everything that goes on, 80% of your genes are under control by what happens in your environment. So just kind of a big picture thing to think about. You know, we talked about the sort of analogy that genes are what sort of load the gun, but the environment is what pulls the trigger. So understand that epigenetics is a huge component to sort of disease progression and so forth. I do want you to know a little bit about some numbers for the Human Genome Project. I know these are on your review sheet there. Some numbers to think about: that there's basically 3 billion pieces of information in your genetic blueprint. Corresponds to about 20,000 genes, and those 20,000 genes really only represent about 1.5% of the total kind of sequence space. So there's a lot of DNA there that's really not doing anything in terms of being translated. And again, we used to call that junk DNA, but we realize it really does serve a purpose, just like your appendix does and just like sleep does. Things that appear to have no known function on the surface probably are really important. So some other things that I kind of want you to be able to think about is that this 3 billion number actually has two contexts. 3 billion is the number of bases that you have in your genetic blueprint. It also happens to be what it cost to sequence the first human genome. And again, it took 13 years. This was not numbers that we sort of talked about here, but it's on your review sheet. So some numbers I want you to know. It took 13 years and $3 billion to sequence the first human genome. And now if we sort of fast forward to technologies like that they use for 23andMe, even though that's not sequencing your entire genome, that actually takes about 2 weeks and about $59. So huge differences in not only cost, but time to sequence genomes. So we did talk about SNPs. I want you to be able to define what a SNP is. So define what the S, the N, and the P stands for. Again, those are single nucleotide polymorphisms. These are not necessarily mutations. They're not necessarily bad. They're not necessarily imparting disease. They are just differences, variations, if you will, found in the DNA between genes of individuals. And so we watched that little video here. Again, I think that was this video here, just a real brief one to kind of highlight that many of these variations, again, don't cause disease, but they just impart different traits. And as we're learning about with some of those traits might have a role to play in disease. And so there's two specific SNPs that we talked about. So all that I really need you to know for the exam, the in-class portion of the exam, is ultimately I want you to know the name of these SNPs and sort of what they're involved with. So for example, MTHFR is involved in folate metabolism. So I do want you to know that it's involved in the production of the bioactive form of folate. I do want you to know the name 5-methylfolate. And again, this relates to the last of our B vitamins, 4 B vitamins you need to know for this test. So again, vitamin B9, which we also call folic acid, is a biological methylating agent. So part of the story that we'll delve a little bit deeper into with sort of a couple of take-home questions are revisiting the story that folic acid is actually an upstream precursor to the bioactive methylating agent, which is 5-methylfolate. So again, folic acid is what you're going to find in most vitamins, but not more and more. You're kind of seeing the bioavailable form, the bioactive form, which is 5-methylfolate. So again, we talked about some applications of this, but not anything you really need to worry about for the exam. You're just going to need to know that MTHFR is the gene that's involved in the production of the bioactive methylating agent 5-methylfolate. The other SNP that we talked about was the ApoE genes. And so these, again, genes involved in fat binding, transport, and breakdown. So there's implications in both liver and brain. Again, we're not going to get into the details of this. This will become part of a take-home question. So when we talk about ApoE, I do want you to know that it's involved in fat binding, transport, and breakdown, and that here are the three different well-established isoforms that we see in the population. So again, ApoE2, E3, and E4. And importantly, the ApoE4 is the one I want you to remember the name of, because that puts you at increased risk for not only Alzheimer's disease, but for traumatic brain injury. So we'll have a lot to expand on in our ApoE sort of story with our take-home questions. We didn't really have a chance to talk about mitochondrial DNA. If I can find some space to fit it in later, I will, but don't worry about anything with mitochondrial DNA. So just kind of going through, take a look at your homework questions as well, because then those again kind of represent a good sampling of the kinds of things you're going to need to know how to do. First question was drawing the structure of ATP. Second was drawing a structure of a trinucleotide. Third and fourth ones kind of get into some structural features of DNA and RNA, why DNA can be more easily separated at high pH. Again, by deprotonating that 2' hydroxyl group, we impart negative charge. Two negative charges that are next to one another in a double helix allow us to more easily separate strands. And again, strand degradation, if we have that, I think I just said a 2' hydroxyl group for DNA. Again, if we have additional charge that we're going to impart, not by deprotonated 2' hydroxyl, but just by deprotonating other functional groups and introducing negative charge that allows us to more easily separate those groups. The 2' hydroxyl deprotonation has to do with why RNA strands are more easily degraded, and that's because we've got an internal nucleophile that can serve to hydrolyze the phosphodiester bond. That's actually chemistry we're going to study for a good bit with our enzyme chapter, learning how RNase A works. Again, thinking about kinase and phosphatase enzymes, we kind of highlighted that in our slide. And then here's just kind of a summary sheet to think about some of the B vitamins that we've talked about. So again, NADH, FADH2, and acetyl-CoA. I didn't specifically put the last one here for MTHFR with folic acid. So we'll see that one. In addition, it's just not on this chart.